All right, good afternoon, everyone. And thank you, Martin, for putting together this exciting session. I'm going to try to give you an overview of ultrasound and what it has done over the past few decades, in fact, and then update you about what we're doing in my lab. So focused ultrasound it relies on having a curved transducer, concave, as you can see on the top left. And you have a geometric focus that can change based on the frequency, and it can be on the order of 1 by 1 by 10 millimeters cubed. And you can see the wavelength lambda there that relies on the frequency. And so changing the frequency, if you lower the frequency, you can bypass the skull very nicely, but you have the longer focus. That's the trade-off. And then when you increase the frequency, it gets more difficult to go through the skull, but you have a tighter focus. So you can play with these parameters along with a lot of other ones that I'm going to try to mention during my talk. So why use focused ultrasound? Obviously, there's so many stimulation techniques, and some of this conference is the showcase of a lot of them. Typically, you have to choose between non-invasive and shallow penetration and poor precision versus invasive with good precision and deep penetration. So because focused ultrasound can be focused in specific region over several centimeters, we believe that it has this unique advantage of having non-invasive, good precision, and deep penetration. So this is definitely not a new idea, and in fact, it's almost a century old. And the idea of using ultrasound waves started with the report by Newton Harvey on frog nerves, where it showed that using these quartz oscillators, which we call now transducers, can, according to different powers, you can actually elicit different spike rates. And later on, a few decades later, the Fry brothers, which are very well known in the therapeutic ultrasound field, had this transducer assembly that you can see there. I don't know if my pointer works. You can see there on the top, on the right. So you, instead of having one transducer, as I showed you in the schematic, you have this 4 different transducers, and they have a focal point in the middle, where all the beams meet, and you have the spike of the pressure, and you can elicit some cortical potentials. In this case, it was actually for inhibition after sonication. And that was in cats, I'm sorry. And I don't know if I can advance it. Oh, what happened? Okay. And then in the 70s, Gavrilov et al. Actually showed this in the humans, in fact, on the fingertips. And then Foster and Winterhold actually showed some auditory nerve stimulation in cats. And then in the 90s, we had this revolutionary idea that Kullervo-Hininen brought together of using MR-guided ultrasound to look at the actual ultrasound beam through the thermal effect that it was inducing inside the brain. And I'm going to talk also about the blood-brain barrier disruption in a second. But also using sophisticated arrays, phased arrays, where you can send different ultrasound waves at different angles and then have them converge and also steer the beam, so you're able to phase the different elements of the transducer and also steer the actual focus. That through the actual intact skull, that was demonstrated almost 20 years back, and it has transformed now to actually using it for essential tremor ablation, FDA approved in the US. So about 10 years ago, Jamie Tyler was able to actually show that if you focus the ultrasound on an excised brain slice, you can actually elicit voltage gate sodium and calcium ion channels activation and trigger snare-mediated exocytosis. So that was one of the first things where LIFU, or low-intensity focused ultrasound, or short for focused ultrasound, or transcranial focused ultrasound, has a lot of different, unfortunately, names, but it all means the same thing, could excite neurons. So in our group, I'm going to show you just 3 studies that we published before, and the first one is actually trying to get higher frequency to match the rodent brain and be able to focus in specific regions and elicit both cortical and subcortical neuromodulation, and both motor and sensory. Then I'm going to show you some more, some target dependence of the AMG characteristics that we're looking at in rodents, and finally show you more on the blood-brain barrier opening and using modulation in monkeys. So our experimental setup is very simple. We have the focus transducer right there on the top. You need the water to mediate, to be able to suppress some of the some of the reflections. The only thing you do to the rodent is to give it a buzz cut. So you just shave the head and nothing else. You put gel to couple the waves to the underlying skull, and then you focus inside the brain through a stereotactic apparatus. And in this case, you can see the EMG cables that we're looking at all the different limbs. And what is missing there is also an infrared camera that's focused on the pupil that I'm going to show you also results on that. So I'm going to focus on this video. You can see the transducer is gigantic compared to the mouse. You see the back of the mouse, the tail, and I have these two blue circles for the two limbs, the hind limbs. And I'm going to play the movie now, and you're going to see the transducer is actually on the left hemisphere, and you're going to see an elicited motor response on the right. And then when we move the transducer to the right, then you will elicit a motor response on the left. So let me play this video now. So the first thing you see that the actual mouse is kicking its leg forward. That's we're on the left hemisphere contralaterally, and then we move the transducer in a second to the right here to the right hemisphere, and then we elicit a similar response on the left. So this is we call this a contralateral response, and you can see this in green on the top, in the bottom right. But we also elicit ipsilateral responses in some areas, and also this is a somatosensory region, which is not necessarily the most intuitive one to get motor responses going. So I'll come back to that in a second. And then when we go to the visual cortex, then we also actually saw a finer motor control. So then again, we're not exactly sure about which circuit we're tapping into, but there's a very small, maybe it's very difficult for you to make it out, but we have a small digit movement. So it's not just a large motor response. We can also elicit very finer motor control, but we're still in the visual cortex there. A few things that we've published before, and that's something that has been shown also consistently throughout the various decades that I mentioned earlier, is that as you ramp up the pressure of the ultrasound wave or the intensity, you actually have two things happening. One, the success rate rises, and we have the plot, the bottom right there, and also the latency shortens. And we show this also with the intensity here on the top right. So you have the latency dropping exponentially almost with the intensity. The rise time also changes in different regions from the posterior to the motor cortex. So the motor cortex has the shorter rise time of the EMG. And then the success rate also increases. So we go from 20% success rate for the responses of the EMG at low intensities to 100% at higher intensities and longer durations of the ultrasound. Something that we also saw was the motor unit firing rate. So we actually saw two different responses for the motor unit. We had the primary motor unit firing rate and the secondary motor unit almost immediately after. So it was a higher threshold for the first primary motor unit and a lower threshold for the secondary. And that's all for motor. For the sensory aspect, as I said, we had an infrared camera focused on the pupil, and we can see that after the locus coeruleus, in this case targeting, we could see a pupil dilation, and you can see the diameter varying in blue during the excitation, as well as the brightness of the actual image. And we could see actually that the responses were varying according to where the focus was. So if we were for the Handley map, we actually had a 1 millimeter depth, and the more sensory responses were happening at larger depths. So a couple of things I want to mention before I switch to the monkey work is that overall we actually have done a lot of safety work. We looked at where we targeted on the left in this side in the region of the somatosensory, and then compared to the contralateral, so we didn't see any damage on H&E. We also wanted to understand the mechanism, and we believe that thermal for stimulation is actually not the mechanism. And in fact, if we look at this, is actually on some of the work that we do on the sciatic nerve of the mouse, and we saw that the maximum temperature there was about degree C, which we don't think is enough according to literature peripheral neurons to excite, but it is actually, in fact, important to suppress or inhibit some of the neuronal responses. I'm not going to show that today, but maybe some of my colleagues may show that. And it is definitely a mechanical effect that we're inducing. And I'm going to show you what happens in this case in the sciatic nerve. So we focus right here where you see red, and you can see a propagation of the motion, in this case, inside the nerve, along the nerve itself. That's a shear wave that's emanating from the actual focus laterally. So we actually try to, we currently imaging that in the brain. We haven't been successful due to some problems with transcranial imaging, but we believe definitely this has a lot to do with the actual response being a mechanical effect. And the last thing I want to show you as far as mechanism is the fact that we actually here elicit sensory neurons in an ex vivo preparation in the mouse, and we compare electrical stimulation to ultrasound stimulation, and the actual potentials smack on top of one another. And you can see the spiking rate that we can elicit. The ultrasound is actually within this red box. It's on during the red box, but you actually have elicitation of the responses of actual potentials even after the ultrasound is off. And we're tapping into some fibers such as A delta and C fibers that meet more latent, where the arrow is pointing, in this case peripherally. So the last part of the talk will be on the monkey work that we're doing. And in this case, instead of just shining ultrasound to the brain, we actually add some macrobubbles that are used as contrast agents in the case of diagnostic ultrasound. It's called contrast ultrasound, to look at, to be able to have more contrast on the ultrasound images. So those are resonators that go into the blood vessels of the brain. And what they do here is that they open the tight junctions, and then there's drugs that can actually reach the neurons. In this case, we actually don't have any injected drugs. We actually allow potentially some dopaminergic agents to reach the neurons, and as I'm going to show you in a minute. So what happens is that we actually can see where the barrier opened. In this case, it will be on the dopaminergic pathway in the mouse. And you can see this is the codeputamen, the substantia nigra. So we open there, the gadolinium infuses where normally the gadolinium would not go through the blood-brain barrier when it's intact. And the same thing we can do in the monkey, very similar to what I showed you in the mice. So we have a stereotax here, and the monkey head is shaved, and then we have the ultrasound going through the intact skull. We can see this in a couple of monkeys here that we did. In blue is where we targeted, in red is where we opened. So very, very targeted and opening there. And we did behavioral testing during anesthesia in this case. So right after opening, during anesthesia, then we had recovered by the anesthesia that there was a touch panel behavioral test where the monkey had to push on a cue, tap on a cue, and then there was a coherence cloud here, and the monkey had to understand in which direction the coherence cloud was actually turning. And there was a reward mechanism. Low reward was 1 drop of water, high reward was 5 drops of water. So briefly, what we actually saw is that you can actually have the initial touch change and actually happen much faster. So you had the response time that was increasing in the contralateral arm. So you were shining on the left, for example, for the ultrasound, and the arm on the right arm will go faster. And we also saw that with the coherence task, you could see a decision accuracy that's actually slightly better, again, in the contralateral arm. And that was actually mainly with a large reward, and the small reward is actually reverse. We didn't see the sonication increasing in this case, but the large reward, it was the reaction time was actually reduced, which actually showed that the motivation aspect was actually changed in this case. And we also now do a lot of awake sonication. So we have the monkey sitting on a chair, and we have the ultrasound continuously going, of course, with pulse ultrasound. The same touch panel is shown here, and we have shown very similar responses, even in an awake animal, where the reaction time is actually reduced. And you can see overall over these hundreds of trials on the top right, you can see that the actual curve is going down, showing that the reaction time is consistently going lower. So in conclusion, we show that the higher resolution focused ultrasound can modulate neuronal activity in different regions of the brain, from motor cortex to somatosensory to visual. Higher pressure focused ultrasound elicits higher pressure success rates and responses. We show that we can affect sensory responses in subcortical regions such as the locus coeruleus. Histology did not show any damage. And we actually see that the successful response rates and mean latencies vary significantly throughout the brain according to the firing of that region. And in the motor cortex, we show that the motor units were capable of firing at frequencies that allowed second motor recruitment. So in the monkeys, through the blood-brain barrier opening, we actually show that you can increase slightly the decision accuracy, response time, and motivation. So ongoing work, we want to explore the parameter space for inhibition and excitation. And as I mentioned, we think that temperature is the key there. We want to integrate fMRI during for our non-human primate studies. So there's a lot of good studies going on also here. So we're getting a lot of ideas. And then finally determine the mechanism behind the blood-brain barrier opening modulation. I want to thank my team, our sponsors, and you for your attention.